I have this because I tried to give you the wrong.
Yes, no reset. All right, so my plan for today is to finish up the last little units of Chapter 5, the structural proteins, and talk about crazy structural protein diseases. Then Wednesday, we'll start enzymes and enzyme kinetics. So you'll start wanting to bring a calculator. If you don't normally carry a calculator with you, you'll want one starting probably Friday. No homework this week. There will be a group quiz on Friday. I do have the exams to get back to you. So overall pretty good. Average an 85. Pretty solid. So good. All right. So typical eukaryotic cells have 3 types of cytoskeletal proteins that form fibers. Fibers is one type of protein. What's the other type of protein we normally talk about? Globular. So globular was the first half of the chapter. This last half is going to talk about fibrous proteins. So we're going to take a quick look at microfilaments. Intermediate filaments. Microtubules, which they are based on the diameter of the fiber. So still really small, on the order of angstroms. But they each have a slightly different job in the cell. Then we'll take a look at keratin and collagen, which are also structural proteins. They're not structural within a cell. They have kind of a different function. Okay, so I'm not going to talk a lot about intermediate filaments. I'm going to do microfilaments, microtubules, talk about tubulin and a few other things. All right, microfilaments. Microfilaments are made of actin. You use them in your cells as a network of microfilaments and specifically to support plasma membranes. This network of microfilaments determines cell shape. And their ability to cross-link allows for increased stability. Okay, so actin and microfilaments. Actin is able to polymerize. When it polymerizes, we then call it F-actin, where the F stands for filamentose. In other words, actin polymerizes to make fibers. Filamentous, something like that. So each one of these little Pac-Man looking guys is an actin molecule. And the actin is going to polymerize as a double chain. So this is a double chain of actin molecules. where each subunit is in close contact with the 3, depending on if that's on an end, or 5 units surrounding it. As you can see from the picture, each actin has the same orientation, so they all end up with their Pac-Mans facing to the left. And the reason for that is because that cleft is where ATP can bind. So we want all the ATP binding sites all facing one way. And then all the non-binding clefts are on the other side, which then makes this side an overall negatively charged end. Which, if this is an overall negatively charged end because of the ATP, by default, this one is more positively charged because the net molecule has to have a charge of 0. And this is also able to polymerize so that more of the actin are going to bind to this end than they are to this end because of just the way the charge lines up. And the polymerization is going to be driven by ATP hydrolysis. So the ATP will become ADP, and that change in concentration of ATP will cause more actin molecules to bind to make the long polymer change. Again, on this picture, we're going to talk a little bit more about it. I have a similar picture on the next page. So again, here's my negative end, there's my positive end. Here are my monomers adding to the positive end. So the polymerization of these actin monomers is reversible, which means they can both add on and they can leave. It's an equilibrium kind of reaction where it can go back and forth. So this polymer double chain is undergoing constant shrinking and growing depending on how many subunits are being added, how much ATP hydrolysis is going on. And they can dissociate from one or both ends of the microfilaments depending on what's going on with that polymer. They typically only add to one side, but they can pop off of either side. And that is going to be called treadmilling. Which is trying to show in this figure here, right? So I have a variety of different monomers. I'm adding some to this side, which makes this side longer, but then this side's gotten shorter. So then in this next figure, more has associated from that side and more has added on. And so the overall position of the chain has shifted to the right. Because here's where it was, now it's here, now it's here. So that would be treadmilling that physically is going someplace. So cells are going to use these microfilaments to grow in one direction and or shorten in another. Which is what allows your cells to kind of, we'll call it move, or at least stretch out in different directions. You're thinking of your biology, looking through the microscope, and the cells seem like they're kind of growing and then like that kind of thing. It's the microfilaments that's allowing your cells to move around like that. And as you can imagine, this system is going to be most used in muscle cells where actin filaments are required for your contractile and relaxation movements. So your muscle cells are going to be the ones that need to be able to stretch and shorten quickly. And actin monomers, microfilaments made of actin, are the reason that happens. Look at that, science. Questions on our microfilaments? Okay, so let's talk about some tubulin and microtubules. All right, microtubules are cytoskeletal fibers built from globular protein subunits called tubulin. These microtubules made of tubulin can assemble and disassemble to allow the cell to rapidly change shape in response to an outside or internal stimuli. So microtubules, which is this over here, are 3 times thicker and more rigid than a single protofilament because it's a hollow tube that's resistant to bending. So once we get to the point that it's a tube, it is much more rigid than a single fiber by itself. Tubulin comes in 2 forms, an alpha form and a beta form, which dimerize. Which they are showing here, that an alpha and a beta tubulin have to dimerize together in order to be able to assemble into the protofilament. The assembly of the microtubule begins with an end-to-end association of dimers, which again is what they're showing here. Alpha, beta, alpha, beta, alpha, beta, alpha, beta. Alpha, betas come together, and they're going to line up always A, B, A, B. And unlike the actin filaments, we can add to both sides of a microtubule. So addition can occur either on the top or the bottom, unlike the one we just looked at where it only really adds to the positive end. This can add to both ends simultaneously. Okay, any other questions? Any questions at all on microtubules? Okay, then let's talk about the stuff that I find much more interesting. Keratin and collagen, which are other structural filaments. Officially, alpha keratin is considered an intermediate fiber. Oh, intermediate filament. So intermediate filaments are structural proteins, just like the other ones we've talked about. Keratin happens to be chemically unreactive, an important component of hair, horn, nails, feathers, and a few other categories. It officially has an alpha helix shape, but because of its overall quaternary structure, it has a smaller coiled shape than what you would think if it's just a regular old alpha helix, right? Alpha helixes have 3.6 amino acids, and they have so much space between each curve. But because it's coiled coil, it's going to be a smaller circumference than what you'd think. So, based on your homework question, these are amino acids number 1 and 4 here on the inside. Are those guys going to be polar, nonpolar, charged, hydrophobic, hydrophilic? I'll take a variety of choices. They're on the inside, and they're only touching each other. Those are our hydrophobic ones, because they choose to associate with each other rather than the outside solution. So then what about my 2, 3, 5, 6, 7 amino acids? Hydrophobic, hydrophilic. Philic. Oh, yeah. So it's going to have 7 amino acids per what's considered one turn before it starts to repeat. Number 1 of 4 amino acids are going to be nonpolar, hydrophobic, because they're on the inside of the structure. I put this on the. Okay. So each subunit, which is going to be one dimer, so one dimer is considered one subunit, where the dimer is this coiled structure. 2 primary chains of amino acids coiled into their secondary structure, which then are associating together as a quaternary structure. Okay? says scratched off.
Oh, okay.
One subunit. Changed my mind about what I was going to say. What part of the dimer would be the termini? The middle or the ends? Ends, excellent. And those are even labeled. N terminal, C terminal. So each subunit termini interact to form a tetramer. How many mers would be in a tetramer? 4, right? So each dimer is going to interact with another dimer to form a tetramer. So then these tetramers, as you can see here, right, here was a dimer, and now I have a tetramer. And this dimer is interacting with that one to form a tetramer. So then those tetramers are going to start forming an even higher ordered structure. So the N terminal and the C terminal are going to associate with the next terminal above it, right? So this N, this, I'll do it in a different color. This N is a C, and that right there is an N. Why would those C's and N's be able to interact with each other if you're at pH 7? Right, we got some charge going on here, right? Is the C going to be positive or negative, typically? The C terminal, negative, positive at pH 7. Typically negative. What about my amino acid end, or my amine end? Positive or negative? Good. What kind of interaction is that called when it's charged? Ionic. We're going to see some ionic interactions. So it's an ionic interaction going on between the terminals. Then, once we have our dimers associated into what these are called protofilaments, they then start to align themselves. Each protofilament aligns with another protofilament. And those two protofilaments align with 3 other protofilaments to make one microfibril. Then, another fun fact. So keratin fibers themselves are cross-linked with amino acids. So they're not cross-linked to the protofilament, it's within the dimers. There's cross-linking within the dimers. And so then they are able to cross-link with cysteine. What kind of cross-linking can cysteine make? Disulfide bridge. So we're going to see some disulfide bridges form between our keratin fibers. Has anyone ever had a perm? One, excellent. The stuff they put on your head, super stinky, right? Typically, super awful smelling. So if you're thinking about getting a perm, which I know are not trendy anymore, they'll probably come back around in a few years. They'll be trendy again. You make that face. I think so. Everything that I did in the 80s and 90s, I was like, there's no way this is coming back again. It is. It's all back. When you wore like the leggings with the really ridiculously oversized sweater, yeah, that was like my 6th grade photo. I definitely remember wearing black leggings and a ridiculously long sweater that came down to my knees. It's back again, except now with a belt. So we didn't have belts then. Or the really tight pants, like ankle, that like get really, really tight at the ankle. Your jeans didn't come that way, so you had to peg them. So you would take the extra material, fold it over, and then roll it up twice so that it would be really, really, really tight at your ankle. That was really, oh, then some slouchy socks. You wear it, that went together real well. So That, and you always coordinate your scrunchy socks with your hair scrunchie so that it put together a whole look. You laugh. Like 5 years from now, scrunchies will be back along with their slouchy socks. So. All right, so when you get your perm, which pretty soon everybody will be perming it up, what it's doing is the first step is to break those disulfide bridges, to oxidize that. And then they'll put your hair up in rollers or whatnot, and then they put something back on your hair to reform those disulfide bridges, to reduce it back to a SS bond. So when you take the curlers out, your hair has been covalently bonded into that shape. Pretty neat, if you really think about the chemistry of it. That some oxidation reduction in everyday life. Again, one of my favorite topics. We're going to get to more oxidation reductions, but pretty awesome. Have you ever thought about why is your hair so stretchy? Like you can kind of pull on it and like it doesn't break. It mostly pulls before it really snaps in half. Some of it's these disulfide bridges, and some of it is the hydrogen bonding and ionic bonds going on in here. These ionic bonds are real stretchy before they're going to break. So you're not breaking a covalent bond, you're typically breaking these ionic and hydrogen bonds. So you're not affecting the alpha helixes, you're not affecting the coil. You are affecting these ionic bonds and the hydrogen bonds between some of these other chains. Fun fact, you can try it later. Pull on your hair, pull on your arm hair, same thing. It's stretchy before it really breaks off. It's all the same hair. I'm assuming that you talk about head hair, but it's all hair on your body. All right, then let's talk about collagen. There we go. Collagen, which is the most abundant animal protein globally. Not saying in you personally, but globally. The most abundant animal protein out there. It plays a major stress-bearing role in all your connective tissues, in your bone, your teeth, your tendons. But it has a unique amino acid composition which gives it a unusual quaternary structure. So I'll talk about this sentence here in a second. Let me give some background. So there's more than 19 types, depending on how you choose to categorize them. 19, 20-ish. Come when we get to number 20, it's very similar to some of the other ones. We're going to say more than 19 types of collagen, and they all have distinct 3D structures. But in general, they all form a rope-like fibrous form. Collagen has a very distinct amino acid composition, which you guys all looked at in your homework, and we're just going to spend some time talking about it. So collagen has a distinct amino acid composition in that every third amino acid has to be glycine. And as a reminder, I guess I'll draw it over. So about 30% are going to be glycines. About 30% of the rest of the structure, so we're going to account for about 60% of our structure, are either proline and or hydroxyproline. So as a reminder, proline's this guy over here. Here you got the carboxylic acid group, but then our amine is wrapped up into a 5-membered ring. Hydroxyproline, what extra functional group do you think is on a hydroxyproline? Hydroxide, which is otherwise known as OH. OH. So what was the funny thing about proline and our alpha helixes? Do they mix? No, bad combination. In general, prolines don't mix in an alpha helix because they are rigid and can't make that tight turn that an alpha helix makes. So putting them into a collagen fiber is a pretty unique choice. It is going to give it a very distinct structure compared to a typical alpha helix. I put this on there. Oh, no, over here. So most often the amino acid sequence in collagen is a glycine, a proline, and a hydroxyproline. What makes glycine one of our unique amino acids? It doesn't really have, it's not chiral, right? It doesn't really have a particularly interesting structure. Proline's this guy. CO minus. All right, that one's glycine, pretty boring amino acid, but tiny. This is one of the smallest amino acids. So glycine only has proton side chains, and both these guys have amino side chains, which constrain the geometry and doesn't make them as flexible. Which means our structure over here cannot possibly be an alpha helix, even though it looks helical. If you're looking at it, it's a little hard to tell, but it's wrapping, the individual fiber is turning left-handed, and then the overall biphyllo, like this darker gray one, is also turning left-handed, as opposed to alpha helixes, which are right-handed. So the high proline and hydroxyproline content is going to force a left-handed helix. Then it's going to wrap with 2 other collagen fibers to make an overall. A triple helix, a triple left-handed helix. If you have high tensile strength, what does that mean about your stretchiness? Super stretchy. So tensile strength is how much force does it take to pull something apart. Right, if you're holding on to both ends of the rope, how hard do you have to pull in order to get the rope to kind of break in half? So collagen requires a lot of energy to do that. So what makes collagen so strong, besides the fact that it has this great triple helix left-handed turn, is that it also is able to cross-link. But of those 3 amino acids, glycine, proline, hydroxyproline, can any of them form disulfide bonds? No, which is why I said can't. Can't form them, right? Because it's only made up of 3 amino acids, none of which have sulfur. So what we're going to do is we're going to cross-link between lysine in some chains and histidine in other chains which is why it's not exactly always the same. And I said typically glycine, proline, hydroxyproline. There are some lysines and histidines that sneak in there. And these lysines and histidines tend to occur at the end of the chains. Does anyone recognize that R group? Lysine, excellent. You had a 50-50 shot. Oh, I've either drawn lysine or histidine. No, that's definitely not histidine. So, excellent deductive reasoning. I'm going to draw two lysines interacting. So that's a lysine. This guy over here is a lysine. The same reaction would occur if it was two histidines or a histidine-lysine combination. It's just a little easier to show when they're both linear molecules. That's not spelled right in presence. So in the presence of lysyl oxidase, based on its name, what do you think that enzyme chooses to do? Oxidizes lysine. Good guess. Lysyl oxidase oxidizes lysine. And what it's going to be able to do, I'm going to just jump ahead to there's a bunch of steps in between. I'm just going to jump in to the end of the oxidation. It forms a brand new covalent bond between these two chains. Tends to be at the ends of the chains. Kind of making one chain link up to the one that's above it or below it. So it turns out that this reaction, this covalent bonding between chains, cross-linking, increases with the age of the mammal that we're talking about. So if you're thinking about this, easiest way to think about like eating beef. Younger beef doesn't have as much cross-linking in it, tends to be more tender when you eat it. Old animals have a lot of cross-linking, so the meat is not nearly as tender because of the increased cross-linking. And cooking it doesn't always help, depending on how you choose to cook it. Sometimes the way you cook it can kind of break these covalent bonds down, but if you just do a quick cook, you're not doing anything. So marinating, same thing. Marinating can sometimes help encourage these breakage of these bonds to try and tenderize a not so great cut of meat. That's the other part, cuts of meat. Depending on what part of the animal, they may have more or less cross-linking. So the beef may or not be as tender as other spots. So then let's talk about I'm hungry, like I'm ready for lunch. So, let's talk about some collagen defect diseases. Okay, I'm going to turn off the video because I want to show pictures, and I currently can't show pictures and my document camera, so